I am Leema Rosemary, Assistant Professor, PG and Research Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. Now I am going to talk about gastric digestion and HCl secretion. HCl secretion mainly occur in stomach. So, stomach is a hollow organ situated just below the diaphragm on the left side in the abdominal cavity. The volume of empty stomach is 50 ml. Under normal condition, it can expand to accommodate 1 liter to 1.5 liter of solids and liquids. However, it is capable of expanding still further up to 4 liters. Parts of stomach. In human stomach has 4 parts: cardiac region, fundus, body and pyloric region. This is the structure of stomach. This is the fundus, body, pylorus. This is the cardiac region. Wall of the stomach formed by 4 layers. First one, outer serous layer formed by peritoneum. Second one, muscular layer made up of 3 layers of smooth muscles fiber, namely inner, oblique, middle, circular and outer longitudinal layers. Then third one, submucous layer formed by areolar tissue, blood vessels, lymph vessels and masonry nerve plexus. Then fourth one, inner mucous layer lined by mucus secreting columnar epithelial cells. The gastric glands are situated in this layer. Inner surface of mucous layer is covered by 2 mm thick mucus. Gastric glands. First one, fundic glands. Fundic glands are considered as the typical gastric glands. These glands are long and tubular. Each gland has 3 parts: body, neck, isthmus. Then cells of fundic glands are chief cells or pepsinogen cells, parietal cells or oxyntic cells, mucous neck cells, enterochromaffin cells and enterochromaffin like cells, that is EC and ECL. Then pyloric gland. Pyloric glands are short and tortuous in nature. These glands are formed by G cells, mucous cells, EC cells and ECL cells. Then cardiac glands. Cardiac glands are also short and tortuous in structure with many mucous cells, and also EC cells, ECL cells and chief cells are also present in the cardiac glands. This is the gastric gland. So it secretes chief cells, parietal cells. It contains chief cells, parietal cells. This is the lumen of gland. Then type of cells and their secretions. First, chief cell. Chief cells produce pepsinogen, renin, lipase, gelatinous, urease. Then parietal cells produced hydrochloric acid, intrinsic factor of Gastrc. Then mucous neck cells produced mucin. G cells produced gastrin. Enterochromaffin cells produce serotonin and enterochromaffin like cells produce histamine. Then functions of stomach. Major function is mechanical functions. First one, storage functions. Food is stored in the stomach for a long period, that is for 3 to 4 hours, and emptied into the intestine slowly. The maximum capacity of stomach up to 1.5 liter. Slow emptying the stomach provide enough time for proper digestion and absorption of food substances in the small intestine. Then formation of chyme. Peristaltic movement of stomach mix the flows with gastric juice and convert into it the semi-solid material, that is chyme. Then other function, digestive function, protective function, hemopoietic function, excretory functions, and many substances like toxin, alkaloid metals are excreted through this gastric juice. Then properties and composition of gastric juice. Gastric juice is a mixture of secretion from different gastric gland. Properties of gastric juice: daily secretion 1.2 liter to 1.5 liter. Then the reactions: gastric juice is highly acidic with pH 0.9 to 1.2, and the acidity of gastric juice is due to the presence of hydrochloric acid. The specific gravity of gastric juice is 1.0022 to 1.004. The next composition of gastric juice. Gastric juice contains 99.5% of water and remaining 0.5% solid. Solid and organic and inorganic substances. The solids are organic and inorganic substances. Organic substances including enzymes and other organic substances. Enzymes are pepsin, renin, gastric lipase, gelatinase, urease. Other organic substances like mucus, intrinsic factor. Then inorganic substances: hydrochloric acid, sodium, calcium, potassium, bicarbonate, chloride, phosphate, sulfate. This is the functions of gastric juice. Actually, pepsin converted protein into proteases, peptones and polypeptides. Actually, pepsin is an active form, but pepsin obtained from the inactive form of pepsinogen. So, the pepsinogen activated by hydrochloric acid. The next gastric lipase. So, actually gastric lipase actively involved in the acidic condition. So, the functions of gastric lipase are: converted dry glyceride to fatty acid and glycerol. The next gastric amylase. This enzyme also act in the acidic medium. Then functions of gastric juice. Digestive function. Gastric juice acts mainly on proteins. Proteolytic enzymes of the gastric juice are pepsin and renin. Gastric juice also contains some other enzymes like gastric lipase, gelatinase, urease and gastric amylase. First of all, pepsin. Pepsin is secreted as inactivate pepsinogen. The pepsinogen is converted into pepsin by hydrochloric acid. Optimum pH for activation of pepsinogen is below 6. Then pepsin convert protein to proteases, peptones and polypeptides. Pepsin also causes curdling and digestion of milk, casein. Then functions of gastric lipase. Gastric lipase is a weak lipolytic enzyme when compared to pancreatic lipase. It is active only when the pH is between 4 and 5 and becomes inactive at pH below 2.5. Gastric lipase is a dibutyrase and it hydrolyzes dibutyrene into fatty acid and glycerol. Action of other enzymes of gastric juice. Gelatinous, it degrades type 1 and type 5 gelatin and type 4 and 5 collagen into peptides. Then urease, it acts on urea and produced ammonia. Then gastric amylase, it degrades starch. Then renin, it's a curdling milk. The next functions, hemopoietic functions. Intrinsic factor of Gastrc secreted by parietal cells of gastric gland plays an important role in erythropoiesis. It is necessary for the absorption of vitamin B12 from gastrointestinal tract into the blood. Vitamin B12 is an important maturation factor during erythropoiesis. Absence of intrinsic factor in gastric juice causes deficiency of vitamin B12 leading to pernicious anemia. Then next function, protective function. Mucus is a mucoprotein secreted by mucous neck cells of the gastric gland, and surface mucous cells in fundus, body and other parts of stomach. It protects gastric wall by the following ways. It protects the stomach wall from irritation or mechanical injury by virtue of its high viscosity. Then it prevents the digestive action of pepsin on the wall of the stomach, particularly gastric mucosa. Then it protects gastric mucosa from hydrochloric acid of gastric juice because of its alkaline nature and its acid containing power. Then finally gastric function is secretion of hydrochloric acid. According to Davenport theory, hydrochloric acid secretion is an active process that takes place in the canaliculi of parietal cells in gastric gland. The energy for this process is derived from oxidation of glucose. Carbon dioxide is derived from metabolic activities of parietal cell. Some amount of carbon dioxide is obtained from blood also. It combines with water to form carbonic acid. In the presence of carbonic anhydrous, this enzyme is present in high concentration of parietal cells. The hydrogen ion actively pumped into the canalicus of parietal cells. Simultaneously, the chloride ion is also pumped into canalicus activity. The chloride is derived from sodium chloride in the blood. Now the hydrogen ion combine with chloride ion to form hydrochloric acid. And to compensate the loss of chloride ion, the bicarbonate ion from the parietal cell enter the blood and combines with sodium to form sodium bicarbonate. This is the HCl secretion. This is parietal cells. This is the part of canalicus. Here carbon dioxide combined with water to form carbonic anhydrase. Carbonic acid is unstable, so it readily converted to bicarbonate and hydrogen ion. Then the hydrogen ion released from the parietal cell to canalicus. At the same time, chloride ion also enter from the parietal cells to canalicus. Here H plus and Cl minus combine to form HCl. Actually Cl obtained from NaCl. So sodium, that is sodium combines with bicarbonate to form sodium bicarbonate. So what are the factors stimulating the secretion of hydrochloric acids? Gastrin, histamine, vagal stimulation. Then factors inhibiting the secretion of hydrochloric acids are secretin, gastric inhibitory polypeptide. Then functions of HCls are activate pepsinogen into pepsin. It kills some of the bacteria entering the stomach along with food substances. This action is called bacteriolytic action. It provides acid medium which is necessary for the action of hormones. Thank you.